Genetic drift is the change in the frequency of a gene variant in a population due to random sampling of organisms. The alleles in the offspring are a sample of those in the parents, and chance has a role in determining whether a given individual survives and reproduces. A population's allele frequency is the fraction of the copies of one gene that share a particular form. Genetic drift may cause gene variants to disappear completely and thereby reduce genetic variation. When there are few copies of an allele, the effect of genetic drift is larger, and when there are many copies the effect is smaller. In the early 20th century, vigorous debates occurred over the relative importance of natural selection versus neutral processes, including genetic drift. Ronald Fisher, who explained natural selection using Mendelian genetics, held the view that genetic drift plays at the most a minor role in evolution, and this remained the dominant view for several decades. In 1968, Motoo Kimura rekindled the debate with his neutral theory of molecular evolution, which claims that most instances where a genetic change spreads across a population are caused by genetic drift. There is currently a scientific debate about how much of evolution has been caused by natural selection, and how much by genetic drift. Analogy with marbles in a jar, the process of genetic drift can be illustrated using 20 marbles in a jar to represent 20 organisms in a population. Consider this jar of marbles as the starting population. Half of the marbles in the jar are red and half blue, and both colors correspond to two different alleles of one gene in the population. In each new generation the organisms reproduce at random. To represent this reproduction, randomly select a marble from the original jar and deposit a new marble with the same color as its parent into a new jar. Repeat this process until there are 20 new marbles in the second jar. The second jar then contains a second generation of offspring, consisting of 20 marbles of various colors. Unless the second jar contains exactly 10 red marbles and 10 blue marbles, a random shift occurred in the allele frequencies. Repeat this process a number of times, randomly reproducing each generation of marbles to form the next. The numbers of red and blue marbles picked each generation fluctuates, sometimes more red, sometimes more blue. This fluctuation is analogous to genetic drift, a change in the population's allele frequency resulting from a random variation in the distribution of alleles from one generation to the next. It is even possible that in any one generation no marbles of a particular color are chosen, meaning they have no offspring. In this example, if no red marbles are selected, the jar representing the new generation contains only blue offspring. If this happens, the red allele has been lost permanently in the population, while the remaining blue allele has become fixed. All future generations are entirely blue. In small populations, fixation can occur in just a few generations. Probability and allele frequency. The mechanisms of genetic drift can be illustrated with a simplified example. Consider a very large colony of bacteria isolated in a drop of solution. The bacteria are genetically identical except for a single gene with two alleles labeled A and B. A and B are neutral alleles, meaning that they do not affect the bacteria's ability to survive and reproduce. All bacteria in this colony are equally likely to survive and reproduce. Suppose that half the bacteria have allele A and the other half have allele B. Thus A and B each have allele frequency 1/2. The drop of solution then shrinks until it has only enough food to sustain 4 bacteria. All other bacteria die without reproducing. Among the 4 who survive, there are 16 possible combinations for the A and B alleles: AAAA, AABA, AAAB, AABB. Since all bacteria in the original solution are equally likely to survive when the solution shrinks, the 4 survivors are a random sample from the original colony. The probability that each of the 4 survivors has a given allele is 1/2, and so the probability that any particular allele combination occurs when the solution shrinks is. The original population size is so large that the sampling effectively happens without replacement. In other words, each of the 16 possible allele combinations is equally likely to occur, with probability 1/16. Counting the combinations with the same number of A and B, we get the following table. As shown in the table, the total number of possible combinations to have an equal number of A and B alleles is 6, and its probability is 6/16. the total number of possible alternative combinations is 10, and the probability of unequal number of A and B alleles is 10/16. Thus, although the original colony began with an equal number of A and B alleles, chances are that the number of alleles in the remaining population of 4 members will not be equal. In the latter case, genetic drift has occurred because the population's allele frequencies have changed due to random sampling. In this example, the population contracted to just 4 random survivors, a phenomenon known as population bottleneck. The probabilities for the number of copies of allele that survive can be calculated directly from the binomial distribution, where the success probability is 1/2, that is, the probability that there are k copies of A alleles in the combination is given by where n equals 4 is the number of surviving bacteria. Mathematical models of genetic drift. Mathematical models of genetic drift can be designed using either branching processes or a diffusion equation describing changes in allele frequency in an idealized population. Fisher model think of a gene with 2 alleles, A or B. In diploid populations consisting of n individuals, there are 2n copies of each gene. An individual can have 2 copies of the same allele or 2 different alleles. We can call the frequency of one allele P and the frequency of the other Q. The Fisher model assumes that generations do not overlap and that each copy of the gene found in the new generation is drawn independently at random from all copies of the gene in the old generation. The formula to calculate the probability of obtaining k copies of an allele that had frequency P in the last generation is then, where the symbol exclamation mark signifies the factorial function. This expression can also be formulated using the binomial coefficient. Moran model the Moran model assumes overlapping generations. At each time step, one individual is chosen to reproduce and one individual is chosen to die. So in each time step, the number of copies of a given allele can go up by 1, go down by 1, or can stay the same. This means that the transition matrix is tridiagonal, which means that mathematical solutions are easier for the Moran model than for the Fisher model. On the other hand, computer simulations are usually easier to perform using the Fisher model, because fewer time steps need to be calculated. In the Moran model, it takes n time steps to get through one generation, where n is the effective population size. In the Fisher model, it takes just 1. In practice, the Moran model and Fisher model give qualitatively similar results, but genetic drift runs twice as fast in the Moran model. Other models of drift if the variance in the number of offspring is much greater than that given by the binomial distribution assumed by the Fisher model, then given the same overall speed of genetic drift is a less powerful force compared to selection. Even for the same variance, if higher moments of the offspring number distribution exceed those of the binomial distribution, then again the force of genetic drift is substantially weakened. Random effects other than sampling error random changes in allele frequencies can also be caused by effects other than sampling error, for example random changes in selection pressure. One important alternative source of stochasticity, perhaps more important than genetic drift, is genetic draft. Genetic draft is the effect on a locus by selection on linked loci. The mathematical properties of genetic draft are different from those of genetic drift. The direction of the random change in allele frequency is autocorrelated across generations. Drift and fixation. The Hardy-Weinberg principle states that within sufficiently large populations, the allele frequencies remain constant from one generation to the next unless the equilibrium is disturbed by migration, genetic mutations, or selection. However, in finite populations, no new alleles are gained from the random sampling of alleles passed to the next generation, but the sampling can cause an existing allele to disappear. Because random sampling can remove, but not replace, an allele, and because random declines or increases in allele frequency influence expected allele distributions for the next generation, genetic drift drives a population towards genetic uniformity over time. When an allele reaches a frequency of 1 it is said to be fixed in the population, and when an allele reaches a frequency of 0 it is lost. Smaller populations achieve fixation faster, whereas in the limit of an infinite population, fixation is not achieved. Once an allele becomes fixed, genetic drift comes to a halt, and the allele frequency cannot change unless a new allele is introduced in the population via mutation or gene flow. Thus, even while genetic drift is a random, directionless process, it acts to eliminate genetic variation over time. Rate of allele frequency change due to drift equals. Assuming genetic drift is the only evolutionary force acting on an allele, after T generations in many replicated populations, starting with allele frequencies of P and Q, the variance in allele frequency across those populations is equals time to fixation or loss equals, assuming genetic drift is the only evolutionary force acting on an allele, at any given time the probability that an allele will eventually become fixed in the population is simply its frequency in the population at that time. For example, if the frequency P for allele A is 75% and the frequency Q for allele B is 25%, then given unlimited time the probability A will ultimately become fixed in the population is 75% and the probability that B will become fixed is 25%. The expected number of generations for fixation to occur is proportional to the population size, such that fixation is predicted to occur much more rapidly in smaller populations. Normally the effective population size, which is smaller than the total population, is used to determine these probabilities. The effective population takes into account factors such as the level of inbreeding, the stage of the life cycle in which the population is the smallest, and the fact that some neutral genes are genetically linked to others that are under selection. The effective population size may not be the same for every gene in the same population. One forward-looking formula used for approximating the expected time before a neutral allele becomes fixed through genetic drift, according to the Fisher model, is where T is the number of generations, Ni is the effective population size, and P is the initial frequency for the given allele. The result is the number of generations expected to pass before fixation occurs for a given allele in a population with given size and allele frequency. The expected time for the neutral allele to be lost through genetic drift can be calculated as when a mutation appears only once in a population large enough for the initial frequency to be negligible. The formulas can be simplified to for average number of generations expected before fixation of a neutral mutation, and for the average number of generations expected before the loss of a neutral mutation. Equals time to loss with both drift and mutation equals, the formulae above apply to an allele that is already present in a population, and which is subject to neither mutation nor selection. If an allele is lost by mutation much more often than it is gained by mutation, then mutation, as well as drift, may influence the time to loss. If the allele prone to mutational loss begins as fixed in the population, and is lost by mutation at rate m per replication, then the expected time in generations until its loss in a haploid population is given by where is equal to Euler's constant. The first approximation represents the waiting time until the first mutant destined for loss, with loss then occurring relatively rapidly by genetic drift, taking time Ni,